welcome to my channel Biology by Katie. If you have seen my videos before, I took a little break from doing YouTube videos due to just being so busy with mock exams coming up and things like that. But if you have seen my videos before, you'll know that I was making my way through the AQA A level biology topics one by one. So we're just going to continue where we left off. So this topic will be, this video will be covering topic 1.5.2, which is DNA replication. As always, I hope you enjoy it. I hope you find it really interesting. And if you do have any comments or any suggestions, please let me know. This video is covering topic 1.5.2. Sorry, I've put the wrong number there. It is 1.5.2, which is DNA replication. So the name of the video is correct. First of all, let's start with a little recap about DNA itself. So on the screen, you can see some information about DNA, aka deoxyribonucleic acid. So this is the nucleotide, the monomer unit. The nucleotide consists of three major components: the phosphate group on the left, the pentose sugar in the middle there, which in this case is deoxyribose, and on the right hand side the nitrogenous base. Now there are four possible DNA bases. There's adenine, cytosine, guanine and thymine. We usually just use the first letter of each to represent these. The complementary pairs, so the ones that will form hydrogen bonds with one another, are A and T and vice versa, and G to C and vice versa. So A to T and G to C and vice versa. You can also see on the screen there the structural configuration of deoxyribose. This image helps us to visualize the position of each of the 5 carbons within deoxyribose, which is really useful when we talk later about how DNA is synthesized. The process of DNA replication is of vital importance in biology. Without it, cell division would be unable to take place. DNA replication is the prerequisite for mitosis, which is the first mechanism of cell division that we look at the A-level syllabus. Mitosis produces 2 daughter cells, each of which are genetically identical to the original, to the parent, and therefore this is a method of asexual reproduction. Mitosis is what allows for the development of new life, as it plays an important role in various stages of embryonic development. Mitosis is also necessary for repairing any damaged or worn out cells in more mature organisms, as well as the progress of the specific immune response. Meiosis is the second mechanism of cell division that we look at that results in the production of gametes. Now, gametes are the reproductive cells of both animals and plants. Cells undergoing meiosis will also complete a full interphase cycle, including the replication of all DNA. This is to ensure that all 4 of the daughter cells produced will receive 50% of the parental DNA, giving them the haploid configuration required for sexual reproduction. Now remember, DNA molecules are made up of 2 complementary strands held together by hydrogen bonds forming between the bases. These strands run anti-parallel to one another, one in the 5 to 3' direction and the other in the 3 to 5' direction. When the mechanisms of DNA replication were first being investigated, it was difficult to confirm which was the right way. The semi-conservative method of replication is the model that is considered to be the most accurate today. The semi-conservative model suggests that the original DNA helix breaks apart and both exposed strands act as a template for new DNA. This results in new DNA that is 50% original and 50% new, hence the name semi-conservative. For this process to occur, we need the presence of all 4 DNA nucleotides, A, T, C, and G. We need the appropriate enzymes that catalyze various stages of the replication process. And finally, a source of energy is required. Energy is stored in molecules of ATP and is released during the hydrolysis of their terminal phosphate group. Let's take a look at the process. So the first enzyme we are introduced to here is DNA helicase. DNA helicase is responsible for breaking hydrogen bonds between the complementary bases on the antiparallel strands. This causes the DNA to unravel and both strands become exposed. This process requires energy from the hydrolysis of ATP. Free-floating nucleotides will then take their opportunity to join up to the complementary bases on the exposed strands. Hydrogen bonds will form between these complementary bases. The number of hydrogen bonds formed between them will depend on which base pair we have. So there are 2 bonds between A and T, between adenine and thymine, and there are 3 hydrogen bonds between cytosine and guanine, C and G. there is a second enzyme involved in the process of DNA replication that we meet now, which is DNA polymerase. Remember from looking at the building of polynucleotides that we have a phosphodiester bond between carbon number 3 on the deoxyribose molecule and the phosphate group of the adjacent single nucleotide. Also, remember that DNA is always a double-stranded molecule and that the two strands run anti-parallel to one another. Now, by anti-parallel, we mean that one molecule runs in one direction and the other in the opposite direction. So let's have a close-up look at this. See how with the strand on the left, the fifth carbon is above the third. So it goes 5 to 3, 5 to 3 on the next, and so on, if we read it from top to bottom. This means that the DNA polynucleotide is running in what we describe as the 5 to 3 prime direction. So this is running in the 5 to 3 prime direction. Now look at the other strand on the right hand side. This has essentially been flipped 180 degrees. We read that strand with the third carbon above the fifth. Therefore, we say this strand runs in the 3 to 5 prime direction. This is why we refer to these strands as being anti-parallel. Now, the reason for this particular feature is that our second enzyme, DNA polymerase, only assembles new strands in the 5 to 3 prime direction. It needs a free hydroxyl group on the third carbon to initiate synthesis of DNA strands. Therefore, DNA polymerase will always attach to the 3' end of the exposed strand and work in the 3 to 5' direction. The new strand is always, therefore, formed in the opposite direction. Because the two original strands of DNA will be anti-parallel, DNA polymerase will travel down each parent strand in the opposite directions, as you can see here. DNA polymerase works by catalyzing the series of condensation reactions between single nucleotides, those single nucleotides that lined up with the exposed bases and formed hydrogen bonds. They catalyze the series of condensation reactions between these to form a polynucleotide strand. So this process ultimately builds the new strands of DNA. I briefly mentioned before that the process of understanding DNA and its mechanisms of replication was not an easy one. There were many incorrect theories before one was actually accepted. Whilst the work of other scientists was credited for early understanding of DNA, it is James Watson and Francis Crick, who you can see there, who are famously credited for their discovery of the helical structure of DNA, the double helix. Their work was pivotal in understanding the operation of DNA, and they both received the 1962 Nobel Prize for their work. Watson and Crick started to give theories about how DNA was actually replicated as well. They came up with two theories, the conservative and the semi-conservative models. In 1958, Matthew Meselson and Stahl carried out an experiment into the action of DNA replication. The experiment and its results seem to support Watson and Crick's semi-conservative model. Now, they based their experiment on the idea that nucleotide bases are nitrogenous. So those DNA nucleotide bases, A, T, C, and G, they all contain nitrogen. This means that the synthesis of these requires nitrogen. Now, element nitrogen has two stable isotopes, nitrogen-15, N15, and nitrogen-14, N14. Now, nitrogen N15 has a slightly heavier mass due to the presence of one additional neutron in the nucleus. This meant that during centrifugation, substances derived from N15 would have a higher density than those made with N14, and that would be observed. So they cultured E. coli bacteria on a medium of N15, so there was only nitrogen-15 present, allowing it to replicate for several generations before extracting and centrifuging the DNA. They did the same with bacteria grown exclusively on N14 medium and compared the results of centrifugation. As expected, the DNA of N14 cultured bacteria was much lower than that of the N15 sample. They then transferred some of the pure N15 bacteria onto a medium containing only N14, and they allowed it to replicate before extracting and centrifuging the new sample. They compared the new DNA to the results of pure N15 and pure N14. The new DNA was found to have a density that was in the middle of the two pure forms. This experiment therefore supported the idea of the semi-conservative replication model, as it showed that the first generation bacteria that was used on the N15 and then the N14 had seemed to incorporate both isotopes of the nitrogen, 50% of the original parent DNA and 50% new DNA. So let's just go through all of the required content from the AQA specification and make sure that we are meeting everything that we need. So the first point is the semi-conservative replication of DNA ensures genetic continuity between generations of cells. So that was where we talked about DNA replication being a prerequisite of both mitosis and meiosis. The next point is that we have to know the process of semi-conservative replication of DNA in terms of all of the above points. So we talked about the process and how that happens and how the enzymes are used at various different points of the replication stage and what roles they play specifically. The final point was we should be able to evaluate the work of scientists in validating the Watson and Crick model of DNA replication. So for this one, we talked about the Meselson and Stahl experiment at the end with the nitrogen isotopes and how that was used as a model to sort of support the semi-conservative method of replication. So we've gone through everything that we need to know for the topic 1.5.2, which is DNA replication. I hope you enjoyed the video.